Good morning. It's 9 a.m. here in San Diego, so we'll begin today's webinar. Thank you very much for joining us and thank you for your support of the webinars. Today's webinar is on MolScreen and in particular the toxicity prediction models inside MolScreen. My name is Andrew Ori, and if you have any questions during the webinar, please feel free to use the chat panel in the GoToMeeting dialog window. If you have any questions after the webinar, please feel free to email or call. And so basically, we have different kinds of MolScreen models. The most basic ones are the fast type models, which are chemistry-based models for activity. This predicted based on big data. So these are ligand-based classifiers, like QSAR models. We also have models for which incorporate the 3D conformation of the ligand, the 3D pharmacophore, basically. So these points represent pharmacophoric properties of one or more chemicals that are known to bind to a particular target. So you can use these to train and build your models. We also have new neural network models, which are very fast. And when data is available, we try and combine as much data as possible into the models. So some models incorporate both the ligand-based and the 3D based. And the DPC models incorporate protein structure from the protein data bank, and also ligand information from the protein database, so the binding pose and multiple structure conformations, and they're basically a docking model. So depending on the amount of information, so obviously some targets don't have enough crystal structures to build a decent DPC model, so you may have a DFC or DFA model, but you can see them in the targets, in the all target table that I'll show in a bit, which kind of models we have. So ADME models that we're going to, and toxicity models will have the prefix MCP and then the target for those models. So ADME tox. 2 weeks ago we concentrated on the movement of a drug through the body, the pharmacokinetics, so what the body does to the drug. So we looked at the different models we have in MolScreen for absorption. So for example, we have models for KCO2 and PAMPA, so predicting how a chemical might get into the bloodstream. Then we have distribution models, so models such as can your drug pass the blood-brain barrier, for example? Does it bind to the P-glycoprotein? Does it get thrown out of the cell, or does it bind to the plasma in the blood as well? So we have models for each of those options. Then we also have models we showed 2 weeks ago, also for cytochrome P450, and how well a chemical breaks down in the body and how it breaks down is predicted. And then we have models for half-life, excretion, and prediction of prodrug from a drug, or vice versa, a drug from prodrug. But in today's webinar, we're going to focus more on toxicity, the pharmacodynamics of the drug. So what the drug does to the body rather than what the body does to the drug. So Paracelsus famously said that all things are poison and nothing is without poison. The dosage alone makes it so a thing is not a poison. So basically, you have to for drug development, we have to find a sort of an acceptable level of toxicity, the dose, the LD50, which is the lethal dose, and you need to look at the narrow therapeutic range, and all of these sort of depend on the therapeutic type. So there are obviously in vitro and in vivo tests to investigate drug safety, as well as looking at toxicity and adverse drug side effects. And in recent years, there have also been some efforts to develop like in vitro models, such as organ on a chip, to reduce the cost of these these assays. However, these approaches are still kind of costly as well as time consuming. So in silico methods, they generally obviously are faster, cheaper, reasonably accurate, and most importantly, you can do them as you're designing and as you're before you synthesize the drug or the molecule in your series. So you can get a good idea of the toxicity levels or the bad groups maybe that you're adding to your molecule that will be flagged. So you can prevent any problems downstream in the development process. So we have different model types for different types of toxicity in the body. So we have a model to identify toxicophores. These are like toxic, it'll flag any known toxic functional group in your molecule. We have models called PAINS, which I'll describe later. PAINS was a concept developed by Jonathan Bale at Monash and his team. And basically we use that for toxicity, basically to identify chemicals that make non-specific covalent binding with other targets in the body, which could be potentially toxic. Then you have tests for carcinogens, obviously the famous Ames test. We have a model called Genotox. That will identify whether your molecule is an alkylating agent or a DNA intercalator. We have models for hERG, which I'll talk about later on, which is a potassium channel. And if you block the hERG, you run the risk of getting arrhythmias. And we also have a panel called TOX21, which is based on the NIH panel of, we have many targets in this panel for common known toxin targets, including like androgen receptor, estrogen receptor, aryl hydrocarbon, and others. I'll show that. So basically for this one, you can just sketch your molecule and run the model, and it will flag which targets your chemical hits. So we'll start looking at the first model. The first model is called the ToxScore, and ToxScore gives a prediction of how reactive or toxic a chemical might be, and it also provides you with a structural alert as well. So it flags potentially toxic groups. The model was built based on 1,000 smart strings associated with toxicity and reactivity, which were collected from various sources. They were assigned demerit scores based on their perceived toxicity and frequency of appearance in approved drugs. The tox score of any compound was calculated by summing up the matching smart strings, and when you run it, a tox score greater than or equal to 1 indicates likely toxicity based on the substructure match. So I'm just going to show it's inside ICM. So if you have ICM, you should have like an icon down here with that you can open, and all these files I use, you can obviously use your own files, or I'd be happy to send you my examples if you want to look at them. So just going to start with. So in this example, sorry. Okay, in this example, we have a docking or screening hit list. We have a number of molecules in our hit list. We have a docking score, so we can rank by score if we wanted to. And we also have some other properties from last week. This is sort of a carry on from last week. We have prediction of pamper, KK binding, polar surface area, molecular weight. But it might be useful to be able to flag the tox score, right, for these leads, so we don't want to be sending chemicals to be experimentally tested that may potentially be toxic. So we can do that by basically go to the chemistry menu to generate the tox score. Go to chemistry and then calculate properties. And you can see that we have inside this panel a large number of properties. We have the ones we covered last week, such as pamper, and we looked at keiko and other things. So there's a lot of the ADME-tox properties are at the bottom. You also at the top we have molecular weight and other things. So further down here we should find the option ToxScore, and it's actually at the bottom here. So you just select that button, that checkbox, and it will run the ToxScore. It takes a little couple of minutes to run on a 1,000 or so chemicals, so I just having the actual results here. So then you'll get a column called ToxScore, which you can sort by the value. And also you have the chemical alerts here. So it shows that there's each of these top scoring, if the value is above 1, it indicates that there may be some substituents that are flagged as being unfavorable or possibly toxic. So if we go down the list, the coloring gets lighter. We have fewer alerts, and obviously these alerts are just for guidance, right? Because some of them you may need if you're designing, for example, a covalent drug. You shouldn't really just immediately filter these out, but it's just a way of alerting you of which substructure may cause problems. So we go down to the bottom of the list, you see that the chemicals get prediction scores of 0, which means they're pretty safe in terms of the tox score. So you can also obviously do interesting things like plot these things. You can filter out. So here we have a plot of polar surface area versus molecular weight, and we just if you right click on any plot, you can change the size of the points or the coloring. So in this example, each of the points in the plot are colored by the toxicity score, the tox score. And so we can quite easily see the larger, the ones with the larger score, and maybe we should definitely remove those. You can filter using the filter tab here in the table. So you can use this to drag to change what's shown in the table. So if you want to lower the filter down to 1 or below 1, then we just have the ones that haven't got a high tox score. In the plot, you can also add things like bubbles. So to show, so when you click on a point, it will highlight highlight that substructure. So you can put mole here, comma, put tox names, go apply. So now we click on a point in the plot, you should see the 2D chemical and the names of the or if you just hover over a point, you can see that information. Okay, so that's ToxScore. That's one approach to looking at the toxicity in ICM. We'll move on to the next slide. So Tox 21, if you're not familiar with it, it stands for toxicology in the 21st century. And toxicology in the 21st century program is a collaboration between several different federal agencies like the FDA, National Toxicology Program, the NIH, and the Computational Toxicology Program as well. And so basically it's a collection of assays and guidance for being careful with chemicals. So the substances assayed in Tox 21 included a diverse range of products such as commercial chemicals, pesticides, food additives, contaminants, and medical compounds. And the goal is really to develop and evaluate and test sort of methods that will better predict how substances may affect the body. So in this program, they have things like in vitro cell assays, They have high throughput screening, sort of robotics methods as well for testing. And they have a set of assays, set of known targets for toxicity. So all of these targets in Tox21 are included into ICM. So you can just sketch your molecule and run it against the panel, and you see like these have hits to these certain targets. Where one is means it's definitely a binder, and then down to 0 being probably not. So in this panel, you have a variety of different targets, but a large majority of them are nuclear receptors. But there's, which are shown in green, but there's, for example, the aryl hydrocarbon receptor in this panel. It's a protein best known for its role in mediating toxicity. So it's basically involved in xenobiotic metabolism, and has long been known to cause immunotoxicity, for example. So there's that target. There is androgen receptors and estrogen receptor in this panel. So chemicals interacting with these nuclear receptors, such as estrogen receptor and androgen. So chemicals they may act as you may be targeting a drug target, but they may also bind to these receptors as off targets. So we need to be careful about that. So or you can also be exposed to chemicals in the environment which may disrupt the endocrine system. So these chemicals may, if they're flagged as being binders to these nuclear receptors, they may interfere with the normal function of the endogenous steroid hormones and lead to adverse health consequences, such as tissue or organ proliferation, reproductive disorders, metabolic disorders, and also cancer as well. So there's also chemicals that do not directly interfere with the nuclear receptors, but they may sort of indirectly interfere. So, for example, anything that might bind to the aromatase, which is a CYP19A1, is an important enzyme for affecting the biosynthesis of estrogen, and so and plays a key role in maintaining the balance between estrogen and androgen in many estrogen sensitive organs. So this is a good one to keep an eye on as well. So this is in the panel. There are also some kinases, some tumor suppressors in this panel that you want to be careful of, not to be an off target. So I was going to show you how you can use this panel. So in ICM, I'm going to delete this. So if you want to try interactively, you can sketch molecules in here in the ligand editor here. You can sketch, or there is a dictionary of drugs here that you can use. But in my example, I already have them prepared. I'm just going to open that example. So I have these 3 chemicals already sketched. If you're sketching them in ICM, you can, as I said, you can use this button here, and then you can append to a table by clicking here, or you can read in an SGF file, smile strings, inchy keys, and other ways of mol format. So that's different ways of bringing this information. But it'd be nice to screen these chemicals against that Tox21 panel. So you find that the Tox21 panel, you go to chemistry, go to MolScreen target panel. So here at the webinar about 4 weeks ago, we went through most of these models and how they work. But you can go back to YouTube and look at those videos if you want to watch more. But go to this panel, and we load the panel of of MolScreen models. So we have a lot of drug targets in this panel. You can load the panel based on like for example, you can load a kinase panel, for example, and just have the models for kinases. Nuclear receptors might be a useful one for this, but all the ADME tox models are at the bottom of this table, and so we can see in this table that we have one for TOX21. So just want to zoom in, you can see the TOX21 panel. It's built on a collection of over 9,000 ligands. You can see the quality of the model in this table here. So basically to run the TOX21 on this table, we will just click on here, check this box, and then it asks us which table we want to run the test on. The table that I read in the SDF file is called test endocrine. You can automatically charge the molecules based on our MolSoft's pKa method. If you have a very large table, you may want to sort of filter that table and just show the actives. And if you click here, it would remove the inactives. But in this example, we'll see that they're all active. But and also it's a smaller table, so you don't really need that option. And then to run the table, we click here, or you can do it as a background job, or you can do it on a cluster using QSub. So I'm just going to press. So here are the three chemicals in our test set, and then we will just run MolScreen. Click here. Should be ready. It's very fast. So we can see that it brings us this table. This is our original table, and it's added a column here. So two of our drugs have a very good profile against the TOX21 panel. So we need to be extremely careful with these models. It's sorry, with these structures, and so it lists all the hits in this column. Or you can so one red is a hit, so you can pan along this table, see the individual predictions for each of the targets that we have. So some of them are, for example, ESR2 here, we have predictions for agonists and antagonists. And if you hover so hopefully you can see this on the screen. So each of these are the target name, the UniProt name for the target here. And yeah, you can go along the table and see your predictions. So the second one is pretty clean. So these two are probably, well, we know they're toxic, but these are hits we need to be careful of. So you may go through your model and notice that maybe you get an unusual hit, or a hit you want to be more closely look at just to be certain. So you can then use the more detailed models. These are fast models based on chemical fingerprints. But for example, we could take the molecule, take one of our chemicals, and then go to our panel. Oh, we've already got the panel loaded, so just go to all targets, which is our panel table, this one. And we can look for, as I mentioned at the beginning of the webinar, there's different models. So there's DFA models, which are ligand-based, but 3D ligand-based. But we'll see if we have a DPC model, which is a docking model for the estrogen, one of the estrogen-related receptor. So it's got ERR1, and the DPC is the docking model, and we can, no underscore. So you can see we have an actual docking model for this receptor. So we can go and run this model, and we get another prediction based on this one includes the receptor structure and 3D templates as well. So you can also double check. So you can run that in the same way. You would select here, run the MolScreen model, and because it's like a docking model, it takes it's about 30 seconds, 40 seconds, a bit too long for this webinar. So I have the example loaded or saved, the results of that, and I just need to find it. This one. Okay, so these are the 3 molecules again that we docked. We can see we have this MolScore. This is against one single receptor. So the MolScore has is above 3. It indicates that this is probably a binder to ERR1. You also get a prediction of PKD shown here. So greater than 6 indicates that it's a submicromolar binder. Probably around 9 would indicate that it's maybe a nanomolar sort of range, KD. And so if we double click here, it loads the complex, the docked complex. I'm just going to move this over here. So you can see that ligand bound to ERR1, so it's very nicely. You also get the docking score, which is called MolRec score. So the lower this score, the better the interaction, predicted interaction. And you can see the power of these models is that they use multiple conformations of the receptor. So if I play this, it's actually been docking that ligand into these antigen receptors. Nuclear receptors are pretty flexible in the binding pocket. So it docks to a combination of those receptor structures. Or you can go down the stack and just look at each of those receptor structures individually, and it ranks them. You can also see for this docked complex, it found a template from the PDB which matched pretty well as well. So it used structure-based docking as well as ligand-based docking in combination. And there are webinars online on how to do that yourself. I can be happy to give a demo on that if anyone's interested. So, so this is what we call the DPC models, which has the docking incorporated. We also have a prediction of lethal dose. So basically the dose required to kill half the members of a tested population after a specified test duration. And so we predict the median lethal dose, LD50, lethal dose at 50%. We have predictions for intravenous, subcutaneous, oral prediction. So in a similar way, it's basically to run the LD prediction of lethal dose, we can just get a new ICM session. We can use, in this case, I'm just going to sketch the molecule. Just going to type, we're just going to use cocaine as an example. We need to charge it. It has a charged nitrogen, auto-assign charges, and then we can save it to a table. So in the same way as the Tox21, we'd go to chemistry, MolScreen panel, and then we can choose the LD50 model here, log LD50, and just run it. It's pretty fast, and you get a prediction for LD50 shown here. So a value of 0 would indicate this like the LD50 is like 1 milligram per kilogram. 2 is 100 milligrams per kilogram. And we're kind of, in this example, we're sort of halfway between those two ranges. So it's a prediction of lethal dose. So the next prediction model I want to show is hERG. So hERG is a potassium channel, and it's sort of essential for the normal electrical activity in the heart. If you have mutations in hERG, you see what you call long QT syndrome, which predisposes individuals to sort of life-threatening fatal arrhythmias. But arrhythmias can also be induced by blocking hERG, and a surprisingly diverse group of drugs can actually bind to hERG. And this is actually one of the most common reasons for drug failure is hERG binding. So it's a good idea to keep an eye on the hERG activity or prediction model as you're designing or screening for new drugs. There is a EM structure map of hERG, which I'll show in a minute. Many hERG binders have similar chemical features, such as multiple phenyl rings. A basic amine group is seen very often in hERG binders, but this is not always the case. So these two, there are other, they can bind very large molecules as well. So this is quite a breakthrough structure as an cryo-EM structure by the McKinnon's group. You can see the paper here. So if we just want to have a look at hERG, we can do that in ICM. So if you read in hERG, the cryo-EM structure has a PDB code of 5VA1, so you can just type 5VA1 here in the PDB search. And when you do, you just get one monomer of the tetramer shown here. So you need to build the full biomolecule. This is the structure from, as I mentioned, it's the single particle cryo. It's an EM structure. See, it says electron here rather than an X-ray structure. And so to build that full biomolecule, you go to Tools, X-ray, Biomolecule Generator, shown here. And that will build the tetramer for you. And you'll see something like this. So drugs bind to the central cavity, the pore region of this channel. And obviously the channel opens and closes. And there are two pore lining aromatic residues, tyrosine 652 and 656. If we look at those, they line the pore of where the drugs or where binders to hERG most likely bind. So obviously there's a tetramer, so we have four. It's a tetramer, so we have four of these groups around the pocket. So there's like a total of 8 aromatic residues lining the pore. There are also 3 residues at the bottom of the pore helix, which contribute to binding. And the pore cavity, this cavity is actually very large. It binds. I'll show you this molecule called MK499, which actually is pretty large. When you convert it to 3D, it's like approximately 20 angstroms by 7 angstroms. So the cavity can bind very large molecules. And we can look at the example now. So it's just if I go file open and then hERG SDF. So these are some known hERG binders, including that MK499, which I mentioned as being particularly big. So to predict the hERG interaction, we need to go to chemistry MolScreen panel load panel. Go OK. And we find the hERG models. There's hERG for classification, so whether it's an inhibitor or not. And there's also a model for predicting the PKD of your hERG binder. So you click here and here. And then you can go just as we did before, run molecules and run MolScreen, and you'll get predictions of hERG. So these will give strong PKD values for these 4 chemicals. So if you hover over, you can see that 6 indicates micromolar, 9 in the range of 9 is most likely a nanomolar binder. Okay. There's also inside ICM there's genotoxic classification. So obviously genotoxic is chemicals that are toxic to the genome, causing cellular DNA damage, mutations resulting in cancer possibly. So genotoxic substances are potentially mutagenic or carcinogenic when inhaled or ingested or penetrated the skin. There's the famous Ames test, which is a very rapid test for mutagens. It utilizes a bacteria that has a mutation in the histidine operon and cannot synthesize its histidine. So the test is to basically see if your chemical can induce the reversion of this mutation and synthesize histidine. So we have the prediction models inside ICM. Common these molecules, like this one shown here, it makes a covalent bond with the DNA, so obviously it prevents replication. These genotoxic compounds are usually acylating or alkylating agents as well. This is shown in 5CR3. This is PDF, it's just a representation of one of them. So, and just wanted to end on pains. So pains is a particularly useful prediction for computational chemists, because these compounds are not only toxic, but they can also just mess up your assays and give out false positive readings. So in ICM, you can use it for your screening data. You can predict pains. So as I mentioned earlier, it's method by Jonathan. It's first described by Jonathan Bale and his team. You can read the papers. This image is from this Nature paper, which gives a good example of some of the common pains like toxoflavin, curcumin, phenylsulfonamides, and rhodonins, enones, and quinones, and catechols are all the worst offenders. Some of these, in terms of toxicology, are that they're covalent modifiers, so they could definitely bind off target to your drug target. So that we need to be careful of these. So we also have like redox cyclers, ones that covalently modify, also complex with metal ions as well. And so these will flag, when we run this on ICM, these will flag each of these groups. So it's a great paper to read if you're not familiar with pains, and I'm going to show it inside ICM. So basically, if we go back to our original screening hit list that I had for the ToxScore, shown here. So we have the score, we had our ToxScore, and then to add the pains prediction, it's a similar way as before. You go to calculate properties, and then here we have mole pains prediction. Just click that, and you will get a column called mole pains, and it flags anything above, I think the cutoff is 0.5. Anything greater than or equal to 0.5 is a pains or promiscuous compound, predicted to be. So you have to be. Some drugs also flag as pains, so you just need to be careful. It's a guide. But less than 0.5 is most likely not a pain. As before, we can just color maybe these points by pains prediction, and we can zoom in to see that prediction. And so you can flag which ones are pains or not. So I'd like to thank you for your attention and for your support of the webinars. I have some questions I'd